Welcome, everyone, and thank you for joining us for today's session on pioneering our understanding of the human brain. It is my pleasure to introduce to you Dr. Bijan Pesaran. Dr. Pesaran is interested in understanding large-scale circuits in the brain and how to engineer novel brain-based therapies. He was trained in physics and made the switch to neuroscience as a postdoctoral fellow. He's been on the faculty at NYU since 2006, where he's an associate professor of neuroscience at the Center for Neuroscience. For more information on our speaker, please look at the Biography tab at the top of your screen. Dr. Pesaran's talk title is Human Brain Science, the Intersection of Translational Neurotechnology and Systems Neuroscience. My name is Dr. Roshana Kashemian, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. We would like to extend our special thanks to the Brain Initiative Program Directors, and in particular, Dr. James Nat, for their efforts in organizing today's session. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting questions during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions in the drop-down box. Our speaker will respond to your questions via email following the presentation. The presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Now, without any further ado, please join me in welcoming Dr. Bijan Pesaran.
I'll be talking about human brain science, the intersection of translational neurotechnology and systems neuroscience. One of the most important things that infuses our work is the idea that the non-human primate is a model of the human brain, provides us with a model of the human brain. And what I'm showing you on this slide is a rendering to scale of two MRI images of the rhesus macaque monkey on the left and the human brain on the right. What's important about this image is that the monkey brain is only 2.5 times smaller in each dimension than that of the human, approximately. That gives us about a 10 to 15 factor of 10 to 15 difference in size. And this is captured by the weight of the two brains, the numbers of neurons in the brains, and the number of neurons in the cortex of each of these brains. And so this is a really important image for us because it shows how similar the monkey brain is to the human brain and allows us to develop our technologies in the monkey in a way that can be directly translated to humans. Another really important aspect to our work is the nature of the signals that we're recording and analyzing. In this slide, what I'm showing you are 3 different recording technologies that are currently used to study the human brain. The EEG, which is shown and measured at the scalp with a 1 centimeter size electrode. MEG, which is measured using a special sensor, which is outside the scalp and even further away. It measures magnetic fields. And inside the brain, with the invasive measurements available in a select population for clinical purposes, we can measure the electrocorticogram or ECOG signal. And that signal is made at the surface of the brain under the dura, and with an electrode that's about 4 millimeters in size. So the technologies that allow these measurements invasively within the brain are shown on the right. They are the subdural cortical electrode, which is shown in the top left panel. It's a grid. And depth electrodes, which penetrate into the brain and are shown below. And each one of these probes has a specificity of around many millimeters and spaced by typically something like a centimeter. When we consider the ability to record neural activity more directly, we need to think about the micro recordings. And so now what I'm showing are a set of recording types that you can make by going into the brain at a finer scale. So I'm showing you the micro ECOG, which is measured at the same location as the ECOG signal, but with a recording electrode that's only 100 microns. And then we have the laminar electrodes and the microelectrodes. And the only federally approved device that can perform these kinds of invasive recordings clinically is the Utah array, and that records spiking and field potential. So there's a big gap between what we can do invasively at the micro scale and at this more macro ECOG scale. So what I want to talk about today is how we can bridge this gap. And I'll start by talking about how to interpret the measurements we can make before moving on to considering the computations performed by the brain and how we can analyze them, and then how we can translate new technologies from monkeys to humans. So interpreting large-scale brain dynamics using field potentials, which is how we record the signals I just talked about, depends on having a model for understanding the measurement. The biophysical forward model is the model which generates from the source signals in the brain the field potentials that we can measure. And that biophysical model is informed by our understanding of cellular biophysics and electricity and magnetism in physics. So we have different types of cells, and we can record the activity on the axons and dendrites, axons shown in red, dendrites shown in blue. And what's important about this model is that it teaches us that when we have, for example, these stellate cells, which are found in layer 4, they have large axonal dendrites, axonal extents, but small dendritic extents. And this type of geometry gives rise to a small field potential in response to synaptic input. In contrast, the other type of neuron that I'm showing you here, cortical neuron, the layer 5 pyramid, generates large field potentials because the axons are not weighted so much by the field potential measurement, and the dendrites are extended, and this is what creates a large signal that we can measure with an electrode. Now, the biophysical forward model isn't the most important forward model to our work. What we also care about is how computations are being performed. And so to understand that, we have to consider what we call a functional forward model. So the functional forward model considers the actual selectivity of neural activity in the brain and how that gives rise to selectivity in our measurements. And so what we have here are two examples of selectivity, which is organized on the left with the arrows showing the spatial coding of a variable involved in behavior that smoothly changes as you move across the cortical surface. And the measurements we make for field potentials in those situations would pool across these neighboring signals and give rise to a large amplitude response in the functional forward model. While if the signals are not smoothly varying in their selectivity across the sheet, then we would get cancellation effectively of the signals and small amplitude selectivity. So an EEG signal might give us a low measurement because it averages over large enough regions of the brain that the topography is lost, while a micro ECOG measurement or a local field potential measurement measured more close to the source, more selectively, may give us a larger amplitude signal. So these ideas are how we think about the interpretation of the field potential measurements. Now, the computations that we're interested in are informed by our understanding of behavior and how the brain controls behavior. In the animal model, in the monkeys, we've been studying the visually guided behaviors. And this is illustrated here, where we have vision entering the brain, guiding actions, and then being used, in this case, to guide looking movement, eye movements and arm movements. Conceptually, the way we analyze these computations is in terms of sensory and motor maps. We think of the eyes as being as seeing the world around us and the sensory cues in that world. And when those cues are to instruct actions, then we need to transform those sensory cues into motor commands or intended movements. And so the transformation that we're interested in goes from the sensory map to the motor map. And in the case of an eye movement, we can think of this quite literally as the map of the world where there are stimuli in it, and then the need to move the eyes to acquire those stimuli. The kinds of behavioral tasks that we can use to study this are correspondingly quite simple. We have a monkey staring at a screen. He has a cue that he remembers the location of. Following short delay, he moves his eyes to that remembered location. And we find that neurons in the brain fire rapidly and in response to the presentation of the cue throughout the delay and then turn off after the movement. As we vary where the cue is presented in space, we find different types of responses from the same neuron. In this example, this neuron prefers or encodes space down and to the right of where the monkey's looking, and the other locations are not as well encoded by the neuron's activity. Now, these neuronal signals are quite hard to record and which impedes our ability to translate technologies based on their activity. Work done in my lab and earlier has shown that field potentials, the field potentials we've just been talking about, also contains detailed information about the processes involving sensory-motor transformations. Here I'm showing you two spectrograms, which show us the power in the signal as the monkey's doing the task, with the cue onset time being shown at C, the saccadic eye movement being shown at S. And we can see that as we move through time and frequency, there are ranges of power in frequencies between 30 to 80 hertz, which are increased when the monkey wants to look to what we call the preferred direction for that site compared to the anti-preferred direction. When we quantify the selectivity of the field potentials and compare it with the selectivity of the firing rate of neurons, we see that they're very similar. The firing rate of the neuron and the field potential power are similarly tuned. So this shows that we can analyze neural computations involved with sensory motor behaviors using field potentials instead of only recording neurons. So how can we apply this to study functions of the human brain that are unique? Well, speech is a uniquely human faculty, and we've been in discussion with in the field. There is this idea that the similar computational mechanisms support speech as these other sensory motor behaviors. And so David Popple, a pioneer in neurolinguistics, advised me to study speech as if a monkey could talk. And the reason for this is that in a model that he and his colleague Greg Hickok have developed, the sensory motor transformations for speech are organized in the brain similarly to that of the other sensory motor transformations in animals. And so this diagram just shows the range of interactions across the two different hemispheres in the human brain supporting speech processing. Conceptually, the idea remains the same. Computational idea remains the same. We have a perceptual map for speech that gets transformed into a motor map that we use to make an utterance, to speak. And the relationship between these two maps and how the computations connect them to go from sensation to action is the type of computation that we can analyze based on our work in animals. And so the strategy for studying speech as if a monkey to talk is to present stimuli that the animal hears, in this case, the human hears, and then speaks based on, in this case, just repeats. And so the task is to listen to the sound and then repeat the word that you hear. You can also have a condition where you just listen to the sound without speaking so that you can hear it, but just to mime it. And the listen, speak, and listen, mime conditions are really important for isolating sensory motor processes. Processes relate to sensation and action, and not just sensation or action. We performed a set of studies to record the ECOG signal in human subjects, epilepsy patients who are having electrodes implanted for clinical purposes, and we find that we can see sites in the brain which respond to the movement to utter, irrespective of whether or not the subject's making any noise. They're not speaking, they're just moving their mouth. We also get other responses which are selective for hearing the sounds, but not speaking the sounds. They can also hear themselves speak in the left panel. And then we have these sensory motor responses, which connect sensation and action by responding both to the hearing and the speech for each condition, even when miming. And so this set of results pointed to the existence of a sensory motor speech system that linked perceptual inputs and motor outputs. And we hypothesized that, along with others, that this interface, this sensory motor interface, supports access to the linguistic processes which we need to ascribe meaning to our speech. The results associated with this study were quite important because they showed, in fact, that these responses were bilateral. That is to say, they were found in both the left and the right hemisphere, which was not expected based on the work of Hickok and Popple and others, indicating that our understanding of how this level of speech processing is lateralized is incomplete, and points to the kinds of new findings we can make about the organization of neural computations in the human brain using field potentials. So now I want to move on to our ability to analyze these neural computations using next generation devices. In particular, I want to talk about so-called flexible printed circuit board based technologies. And the reason why is because they're really powerful for translational goals. These devices are microfabricated, they're cheap and low cost, they're available for manufacturing commercially. They are also able to be used to make biocompatible implants. And critically, since they're based on printed circuit boards, we can integrate more advanced electronic circuits that make the devices wireless and smart. So we've done a number of studies to test these devices in our monkeys. The way we've done this is to revise how we think about the testing cycle. Instead of pairing and training animals and then implanting a device and seeing how it does, revising the device and then implanting it again in a different animal, we developed a system whereby we could package the device, shown in panel E, and then place it inside an implant, shown in panel D and F, that allows us to prepare one animal and then implant many devices rapidly. This is because we don't need to re-implant the animal surgically. We can do it while they're awake. We've also used this system to record wirelessly overnight. And here I'm showing you what we call it's a cage system that has wireless antennas on it. And then we added a wireless transmitter to the previous assembly that I showed you. What recording these signals overnight allowed us to do is to study for the first time these signals in naturally sleeping monkeys. And what we can do is look at the sleep dynamics. So this is a different kind of activity than what we've been talking about, but illustrates the power of this approach of recording the micro-ECOG signals on those electrodes. And so on this slide, what I'm showing you are the different types of phenomena that we've observed, where we see signals which are present both on the spiking of the neurons, the field potentials inside the brain, and on the ECOG near and far. And that's in the global example. It's present on all the contacts we recorded from in this experiment at the same time. We also, however, see other kinds of responses which are local, which is to say they're on the ECOG and they're on the LFP, but they're not on all the ECOG channels. There's a blue, the far ECOG is silent in this case during the event. We can also find other events which are more found on the field potentials than on either of the ECOG signals. And finally, we can see events that propagate across the cortical sheet and, in fact, from the depths of the brain to the surface of the brain. All of this is to show how well the micro-ECOG signals can resolve high-resolution signals in sleeping brain, which has previously only been studied using more macroscopic measurements like EEG that do not resolve these different kinds of responses. Most recently, we've been taking these devices and testing them in patients with epilepsy again. But instead of recording the ECOG signal, we're recording the micro-ECOG signal using our new device. Here, I'm showing you intraoperative recordings of the micro-ECOG signal during seizure-related events. And this shows about 100 of the electrodes that we're recording from. And you can see as you go down through these different electrodes, which are spaced quite closely, the activity is changing across these different contacts quite dramatically. You see events coming and going as you move across the sheet. And now I want to show you a video of this data, the same data that I'm showing you in the time series, but now as a movie. And you'll see the different colors showing the different voltages of the signal over time. So what I hope you can see in that movie is that the activity across all the different contacts is swirling and very dynamic and rich with structure in ways that we could not have observed if we had not made the measurement the way that we did. That is to say, with high resolution using the micro-ECOG electrode array. And this is really important for clinical applications because the different types of events need to be diagnosed differently, and the treatment that the patients will receive should reflect this diagnosis. So in conclusion, I've told you about how field potentials can be used to interpret and understand the organization of brain activity, and the functional forward model is a very important concept for this because it allows us to interpret the selectivity in the signals and how we use them to understand neural computations. I've also shown you how we can develop a framework for analyzing neural computations that's been used to study visually guided behaviors in animals, and to do so for what is a uniquely human faculty, that of speech. I have presented how we can translate new technologies that are low cost and high resolution that will allow us to resolve dynamics in more detailed ways than we could before and offer new treatment paradigms, we hope. Thank you very much.
Thank you, Dr. Pesaran, for that excellent talk. As a reminder, our speaker will follow up any questions about his presentation via email. Continuing with our innovative neurotechnologies track, we have the outstanding Dr. George Buzaki presenting next. Dr. Buzaki's talk title is High-Resolution Recording and Non-Invasive Perturbation Tools for the Human Brain. Hope to see you there.